Hi class, hope you are doing well. Welcome to chapter 3, part 2. We'll actually be talking a little bit more about chapter 3 in terms of cells here very soon, but I kind of just want to give you a little extra part to sort of break this down because chapter 3 can be a little bit daunting. So today what we're going to do is we're going to be talking about movements of the cell. So remember that the cell has a cell membrane. And so things can either go into the cell or out of the cell. But remember that it has a phospholipid bilayer, meaning that not everything can pass in and out of the cell as easily as we'd like. That's why we need proteins. But when we're just talking about movements in and out of the cell, we have two basic processes. And so we think of something called passive or active transport or active process. If you're thinking about passive transport, it doesn't actually require energy in order to make this work. And the reason is because things move from an area of high concentration to an area of low concentration. Meaning that hypothetically, I'm going to give you as an example. If you were making cookies today, your kitchen's going to smell fantastic and it's going to have that wonderful aroma of cookie smell. But what happens is that gas, that smell, will not only stay in the kitchen, it'll actually linger maybe into your dining room, your living room. And that's a passive process that's happening. Same thing actually happens with a cell as well. There are things that are going to want to travel from an area where there's really high concentration to an area that there's not so high. And the reason is ultimately is to have it be equal. Now, in the cell, we don't allow things to be equal, but this is the process that it would take. And to make this happen, it doesn't require any additional energy. When we hear the term ATP, think energy. So in this process, we have something called diffusion, which is diffusion of gases, osmosis, which is the diffusion of water or the passive transport of water. We have something called facilitated diffusion, in which proteins will be used. And then we have a process called filtration, where we'll actually be filtering our smaller components from our larger components. And we'll talk more about those in detail here just in a moment. Now, besides passive process, we also have another basic process called active. Usually I call them passive and active transport. Now, active transport is on the opposite idea. Imagine that you're starting off in an area of low, and you're going to be going against this concentration gradient to throw things in an area of higher. So for instance, if you had more sodium on the outside of the cell than in, imagine that you're taking more stuff and throwing it outside of the cell. So where there's already an area of higher, you're making it even larger or making it more concentrated. So because you're going against that concentration gradient, it requires energy to make this happen. Kind of imagine, if you will, that it's like you're trying to swim against the current. It requires energy to make that happen. Well, we have processes that are going to do that. So when we talk about active transport, that's what's actually happening. And in terms of active transport, we have different movements inside and outside of the cell, what we refer to as endo and exocytosis. And then there's a transcytosis that's sort of a mixture of the two in between membranes. So we'll be talking about those here in just a moment. All right. So thinking about that passive idea, imagine, if you will, that you have a sugar cube. And again, that sugar cube is really highly concentrated in one particular area. So that's your area of high concentration. But throughout time, what's going to happen is those sugar molecules don't want to stay in this area of high. They want to move to an area of low. And eventually what they're trying to do is hit equilibrium. So where it would be equally distributed all throughout this glass. Now, again, in the human body, we don't allow for that to happen. We do want there to be different concentrations, and we want areas that are higher than others. But this is the goal that happens with this passive transport. So with diffusion, it's the movement of atoms, molecules, or ions from an area of high to an area of low. And again, the reason is trying to go from a really highly concentrated area and then slowly be moving them to hit that equilibrium where it's equally distributed. And again, because of how this happens, it's a passive process, so no energy is actually required. And so we'll talk a lot about this when it comes to moving of oxygen and carbon dioxide, as well as other lipid soluble substances too. And so here's a nice little picture to be able to see as well. Here, what we're looking at is a beaker of solutes, which we're going to refer to just as salt. We'll make it salt. So that's what we see in red. And then we have water molecules. And there's a membrane in between them that's permeable to both. So again, throughout time, you'll see that figure A has the most red solutes. And so that's the area of high there. And in B, you have a lot of water. So what's going to happen is throughout time, those concentrations are moving from areas of high to areas of low. And eventually, again, that equilibrium would be hit or reached. Now, facilitated diffusion works the exact same way, but keep in mind, now we have a little bit of a barrier. So as I told you guys before, when you think about the cell membrane, you have these hydrophilic heads and these hydrophobic tails. And because you have those hydrophobic tails, not everything is allowed to pass through. Lipid soluble membranes and very small gases can pass through, but if you're a sodium ion, if you're glucose or an amino acid, you cannot actually pass through. So what happens is there is a protein carrier that will allow for that movement to take place. It's still going to go from an area of high to area of low. It's still passive because it naturally would do it, but now you just have a barrier. So because of that, we get a little help from our friends. That's why we call it facilitated diffusion. It's still passive transport. It's just allowing for that protein to be able to actually allow for that passing to take place. So we still are moving from an area of high to an area of low. We're just needing a little bit of help. So with facilitated diffusion, we get by with a little help from our friends. And so we can either do this with a carrier or we can do this with a channel to make this take place. But basically, I just want you to know the nutshell of it. With facilitated diffusion, again, it's because it's still going to move it down the concentration gradient, area of high to low, but because you have that phospholipid bilayer, you need to use proteins in order to make this take place. And with channel-mediated facilitated diffusion, there's different types that we'll mention. I will not go into crazy detail about this, so you can sort of disregard this. All right, now to give you a better picture of sort of what to expect when we turn in terms of facilitated diffusion. Again, it's helping with ions, it's helping with glucose, it's helping with amino acids. And again, what you can see here is this protein channel that's in purple is allowing those substances that normally wouldn't be able to cross to be able to get in. So we still have this area of high, area of low, but now you just see it in a little bit more detail. All right, next passive process that we have is going to be osmosis. Osmosis is the movement of water molecules. So that's going to be the hint to know is the movement of water molecules from an area of high to area of low. This is actually extremely important, especially when we start talking about the human body and thinking about volume regulation and blood pressure and things like that. We actually talk about osmosis quite frequently. We may not think about it from that perspective, but that's what's going on. So what you're seeing over in this picture is that in beaker 1, you have these protein molecules and they can't pass through that membrane. And then you're going to see that you have water molecules. Now, what's going to happen is if you have more protein on one side than the other, because of the fact that you now have this change in your solutes, water wants to move in the area that's more higher concentrated. And my little way to remember this, I'm actually going to text it or type it out, is where salt goes or solute goes, water follows. That's the way to remember this. So where there's a higher concentration of salt, where there's higher concentrations of proteins, where there's higher concentrations of solutes, water wants to move towards that. And again, it's to equal it out. And this is a passive process, no energy is required. But again, what you're able to see in this beaker is now that movement of water is going to shift to try to dilute out that other side. So we really talk about this a lot in terms of blood pressure. So if you think about it, if you had a bag of chips and a glass of beer, or maybe like something very salty, like a pretzel or something along those lines, very full of salt. What happens is your body wants to retain water because of that, due to this osmosis. Because of the fact that you're trying to equalize the amount of salt that you have, you're trying to sort of dilute it. And so therefore you retain water. If you retain water and you're not peeing that out or excreting that, it increases your blood volume, which therefore will increase your blood pressure. So sometimes you may hear about patients, or maybe someone that you know is on a water pill. That's what they refer to as. It's called a diuretic, and it helps for patients to actually lose that water. So that way they can restore their blood volume and therefore restore their blood pressure. And so again, where salt goes, water wants to follow. So I hope that makes a little bit of sense. And I'm going to bring up a couple more ideas for you to think about. All right, so because of this really big play between salt and water, and again, for the human body perspective, we spend a lot of time dealing with this. And again, because not only do we see it from a cellular level, we can see problems at an organ and organ system level. So we worry about something called osmotic pressure. And it's actually this pressure that happens between the cell, and it's going to increase or decrease depending on the amount of solutes that you see. So in figure A, you're looking at a red blood cell. Now, a red blood cell has a certain sort of shape to it. It's called biconcave, kind of looks like a little bit like a donut. In an isotonic solution, and iso means the same. Tonic is thinking of pressure. The amount of solute that you have equals the amount of water or solution that you're going to see. So there's no water entering the cell or losing of the cell. It's pretty much maintained. There's no changing that's taking place. Now, in a hypertonic solution, the outside of the cell, imagine, is full of salt, like salt water, if you will. Like you put this poor little red blood cell in salt water. What happens here is because there's so much salt outside of the cell, water wants to follow, and it's going to leave the cell. Because of the fact that it's leaving the cell, that red blood cell now is going to shrivel up. On the flip side, if, let's just say hypothetically, you put this red blood cell in pure water, where there were no other solutes or salts, now that water wants to go inside, because where there's higher salt, where there's solutes, water's going to follow. And now the water is actually going to swell up the cell, and that cell may actually burst. So we have to be very careful with that in the human body. We do not want that to take place. We do not want to have our cells shrivel, and we do not want to have our cells swell. So we have to be very careful to monitor this component. And so the way to kind of remember this, to make life a little bit easier when you're trying to think about what's going on, in a hypertonic solution, remember that like hyper people tend to be smaller or skinnier. It's not always the case, but it's a good mnemonic way to remember it. And if you see hypo, think of hippo. So they're going to be bigger. A student told me that once, that's how she remembered it. And so ever since then, I kind of just use that as a reference. But again, when we're thinking about this, again, that's what's kind of going on, is we have this changing of water in and out of the cell. And so if you're going to be losing water, you're going to shrivel up. And again, the reason they're losing water is because there's more solute outside of the cell in a hypertonic solution. In a hypotonic solution, there's not a lot of solute outside of the cell. And so therefore the water wants to flow in. And that's kind of what's happening here. If you see hypertonic, it means excessive pressure. And again, we're thinking of the solution. And if you see hypo, it means deficient. So you're not thinking about what's happening inside the cell, you're thinking about what's going on in the solution. But again, if you remember hypo and hippo, that may also help you as well. All right, and then the last passive process that we're going to talk about is filtration. So filtration is actually a force that happens that is going to force molecules through the membrane because it exerts a pressure on it. So from here, this helps you to separate your very large particles from your smaller ones. And so one of the examples that we talk about is that when your blood plasma, the fluid part of your blood, actually leaves the capillaries, the blood vessels, the water and the small solutes are filtered out, but your big proteins stay inside of the capillary. So this is a filtration process. And again, it's passive. There's no energy that's required to make this happen. Now with active transport, you are going against that concentration gradient. You're going from an area that's already low to an area that's high. And because again, you're going against this concentration gradient, imagine that you're going against the current. So because of that, it requires energy to make this happen. So one example that we like to give is actually something called the sodium-potassium pump. In our cells, we have a lot of sodium outside of the cell in the extracellular fluid. On the inside of the cell, in the intracellular fluid, we have a lot of potassium. And so normally what wants to happen is sodium wants to flow in and potassium wants to flow out. But we have this pump that actively makes sure that that doesn't happen. It basically is like, oh, no, you don't. And it pulls the potassium back in and it throws the sodium back out, basically makes it leave. And so because of that, that's considered to be active transport. Now, part of active transport is we also have endocytosis. So endocytosis is the movement or substances inside of the cell. And so we'll talk way more about this coming up. But there's 3 different types of endocytosis. Pinocytosis is actually, if you think of Pinot as wine drinking, it's engulfing liquids. For phagocytosis, phago is thinking of food, so thinking of engulfing solid particles. And then we have some that are receptor mediated. So when this substance actually is bound to a receptor, then it engulfs it and brings it inside of the cell. So we have 2 different types of endocytosis that you'll really hear a lot about: pinocytosis and phagocytosis. Receptor mediated, you won't hear about a lot, but again, just know it's because it's bound to a receptor on the membrane, and therefore then the cell engulfs it. But Pinot, think of wine, think of drinking. So the cell is engulfing liquids. And with phagocytosis, it's engulfing particles. And again, this is via endocytosis. You're bringing things inside of the cell. We also have exocytosis as well. And with exocytosis, they are actually leaving the cell. You have the opposite approach where you're using vesicles to release contents out of the cell. And so one example that we'll talk about is this is actually what happens when you release neurotransmitters. You may have heard of something called acetylcholine before. So when we release that neurotransmitter to the cells, we actually undergo exocytosis. It's leaving the cell. Or maybe if you guys recall from our previous lecture, when I showed you the Golgi body, how it had those vesicles that were leaving and actually transporting substances outside of the cell. That was via exocytosis. And then transcytosis is actually a mixture between endocytosis and exocytosis. So what happens is it quickly transports a substance from one cell to the other. And this is actually what happens when you think about the HIV virus. So what you can see here is that you have these HIV infected cells, and they are either going to be, in this circumstance, we're looking at the anal or vaginal canal. They're going to bud and they go through this receptor mediated endocytosis. So their substance is going to bind to that receptor and it engulfs it in. Now what's going to happen is it moves across this membrane and then it undergoes exocytosis to leave. Now, when it does this, now it's able to interact with our white blood cells. And one of the things that the HIV virus does is that it actually infects our white blood cells and it causes issues with something called our T cell levels, and it affects immune system response. So a lot of patients actually, or I should say, patients, especially like in the 1980s, one of the reasons that the death was so high was because they didn't have a way to stop this program from happening. They didn't have a way to block this, but also, the issue is the fact that their immune system was so low. So even like a common cold or pneumonia would actually be the fatality. So this is the transcytosis. It's this receptor-mediated endocytosis following exocytosis. More than likely, I'm not going to test you on this, so you don't have to worry about this. This is more of FYI, but I like to make mention of it. Now, what we do have to keep in mind is the fact that we have these cells, and cells are going to have to reproduce. So when you think about it, we have one cell, which is going to be our parent cell. And what's going to happen is it's going to undergo something called cell division. So instead of having just one parent cell, you end up with two identical daughter cells. So when we think about our cells replicating, for instance, your epithelial tissue, like your skin, your lining in your mouth, those are going to replicate very easily. Some cells do not. For instance, like your nerve cells really don't replicate. Your cardiac cells really don't replicate, but other cells can. So if we're thinking about growth and development and, growing more muscle mass and tissue, we have this replication that's going on. So we go from one parent cell to two identical daughter cells. And it's through this process called cell division. And so when we think about this cell division, sometimes you'll hear it called the cell cycle. So the cell cycle is a series of changes that happen. So again, you'll start with one parent cell, and at the very end, you end up with two identical daughter cells. Now, in this picture, I want you to keep in mind that a lot of cells are not always replicating. Our cells aren't constantly, generating over and over. We have certain cells that do, but some of them may not be undergoing this. However, if they are needing to replicate, there are stages that will occur. The first stage is called interphase. And so interphase is actually this entire component. The majority of time that is spent in the cell cycle is actually in interphase. We don't give interphase enough credit, so I'm going to do it for you guys. Interphase is going to be where the cell is actually growing. It's going to reproduce its genetic material and make a copy of it. And then it's kind of double checking to make sure that the cell is ready to go before it undergoes something called mitosis. And mitosis is where the cell is now gearing up for that cell division. And then finally, the cell will undergo something called cytokinesis, meaning that it goes from having two cells that are just held together to actually having that cleavage and separating it into two daughter cells. Now, for the cell cycle, I'm going to go over the components of mitosis. I will not test you on it specifically. So you do not have to know every little component. Even if there's some maybe homework questions that pop it up, you will not be asked specifically about knowing every little bit about mitosis. But I want you to have a general understanding of what is going on in the cell cycle. So the majority of time, again, if they're in the cell cycle, the cell is actually spending it in interphase. It doesn't spend that long in mitosis in comparison, but the mitosis is what we end up memorizing and knowing in so much detail. So I like to bring more attention to the interphase, talk to you about how the cell is gearing up to divide, and then finally, again, how the actual splitting takes place. All right, so in interphase, this is a very active period. The cell is growing. Imagine that you were doubling in size because, again, you're now going from being one parent cell to actually splitting into two identical daughter cells. So the cell has to still maintain its normal functions, but it's also replicating the DNA. It's preparing for this division. And so it's making more of itself to prepare for that. And so when we talk about the phases, there are G1 and G2, which are called the growth phases. And so again, they're replicating their structures, they're helping to grow bigger. And then finally in the S phase, this is where our DNA is being replicated. Now, bringing back some information on this. DNA is, again, the genetic blueprint. It's found in the nucleus. And if you guys remember the nucleus, it contains the chromatin. So the chromatin is going to be where our DNA is found. And so we have to make a copy of that to make sure that that is replicated so that each cell has it correctly done and they have all the components that they need to be able to maintain normal functions and carry out daily activities. So again, for our cells, it spends most of the time in interphase when it's undergoing this cell cycle. Now, in terms of mitosis, this is the division of the nucleus versus cytokinesis is the division of the cytoplasm. There are different phases of mitosis, and the way, if you were ever to be asked this again, the way to remember this is I passed my anatomy test, prophase, metaphase, anaphase, and telophase. These are the phases of mitosis to actually start doubling the cell and getting it ready to be split into two identical daughter cells. Like I said, I won't be testing you specifically on this, so you don't have to know the crazy detail behind it, but I just like to make mention of it. In prophase, you're going to have the chromatin from the nucleus condensed to form chromosomes. So remember, if you will, that the chromatin is like almost like yarn when you see it in the nucleus. During mitosis, it actually condenses and makes these little X's, which we refer to as chromosomes. Centrioles, which if you remember from before, those really important little organelles, they are going to actually help to move to opposite sides. And we're going to notice that the nucleus kind of starts to disappear. In metaphase, meta I always remember is straight or middle, we have these spindle fibers from the centrioles that actually attach to the chromosomes, and they line up in the middle of the cell. In anaphase, we now pull our chromosomes apart and they move towards different opposite directions of the cell. And then finally in telophase, the chromosomes start to return to chromatin, our nucleus comes back to normal, and we start to notice that we kind of have two cells. They're just going to be conjoined at the middle in this one particular portion. But otherwise, they're pretty much two functioning cells that just haven't divided by the cytoplasm yet. And so those are the components that we end up seeing. So in this picture, this gives you kind of an idea of what to expect. This is our prophase. So again, in this picture here, you can see that you have a nucleus with that nuclear envelope. We still have chromatin, and we got those little centrioles that are going to help with this division. In prophase, now we can see that those chromosomes condense that chromatin condenses into chromosomes, and our nuclear envelope starts to disappear. Then finally, when we start to look at metaphase, they are starting to line up. We see these chromosomes starting to line up in the middle. And what's going to happen in anaphase is they actually pull them apart. Because what's going to happen is one side's going to take this stuff, one side's going to take this stuff. And then when we go to look, we can start to see that in telophase, they're starting to kind of function into two different cells that are kind of just conjoined by this cytoplasm. And so what you see here is that you have one daughter cell here and one daughter cell here. They're just joined at this little cleavage furrow. And then finally, what will happen in the cytokinesis is they will actually separate, being two identical daughter cells. So like I said, I won't be testing you specifically on each individual piece. I just sort of want you to have a general understanding of what's taking place in the cell cycle. All right, and then finally, again, with that cytokinesis, this is the true division that happens. So it starts to happen during anaphase, and then it kind of runs through telophase. And what you're going to notice is that this is the true almost pinching, if you will, and pinching the cells in half. So again, they're kind of conjoined at this little cleavage furrow where you have now two operating cells that are just kind of conjoined together. And then finally, in the cytokinesis, they're finally separated into two functional cells on their own. And so what I want you to take from that is the fact that cell division is very important. So remember that interphase is sort of gearing the cell up for the cell division. The mitosis is the true, like actual getting ready to cell divide and division of the nucleus. And then finally in that cytokinesis, we're now truly pinching them off and becoming two identical daughter cells. So the frequency of cell division is very much regulated. So again, your skin cells, your intestinal cells, if you think about the cells in your mouth, they are constantly dividing often, and they're continuously doing that. You constantly are making new skin cells. You're constantly making new blood cells. But cardiac cells, not so much. Neurons, very rarely. And so because of that, again, our cells are very strongly regulated, which is a good thing and doesn't allow for this to happen all the time. And so when we think about our cells dividing, they actually are very much regulated even by their surface area as well, meaning that if cells start to crowd each other, they don't want to divide anymore. They don't want to kind of overlap on top of each other and divide and not have room and not have resources. So cells actually divide in more favorable relationships in that way too. And we have constant controls, whether they be hormonal or growth factors, that are really controlling the cell division. So when you hear the term contact inhibition, healthy cells actually stop dividing when they become crowded. Now, the funny thing is, like cancer cells actually won't. Cancer cells, like when you see a tumor, they lose that contact inhibition and they just keep growing and growing and growing until you end up developing this large mass or this tumor. And so I just wanted to make mention of that to sort of talk to you guys about those ideas. I hope that makes a little bit of sense and kind of brought to you a little bit more about the cells and sort of how they operate and how they work. Thank you so much for listening, and I hope you guys have a wonderful day.